session. After that, we have a keynote session in the evening at 6 o'clock. So this is a session on cancer immunology, and no other than Professor Shubhda Chiplankar is the moderator. You all know her very well, but I'd like to introduce her once more. Shubhda Chiplankar is a former director of Advanced Center for Treatment, Research and Education in Cancer, which is also called ACTREC, very commonly associated with Tata Memorial Center, Navi Mumbai. She earned her PhD from University of Mumbai in 1983 and postdoctoral fellowship from Max Planck Institute in Germany from 84 to 86. She has over 35 years of research experience in the field of cancer immunology. In fact, she carried on from where Professor Sudha Gangal, she was her mentor and guide, and I know her because she was very close friend of my mentor, Dr. Shobha Sehgal. So today we had Dr. Malviya also. They are of the same group. So I know the legacy. She is member of several scientific review committees, task force committees, and academic council of universities. At present, she is the member of the education committee of International Union of Immunological Societies, the IUIS, and president of Immuno-Oncology Society of India. Her research interests are understanding the immune dysfunctions in cancer patients and development of immunotherapeutic strategies for cancer using gamma delta T cells. So, ma'am, I hand over the session to you for moderating, please.
Yeah.
Over to you.
Thank you, Sunil. Though I'm not visible, and I'll try after some time whether my video works. Thank you very much for the invitation. It's a pleasure to be here. Last year we organized the FIMSA in Jaipur, and this year you all have organized. It's a very nice program. And today we are, this is the session related to cancer immunology. And we have two very eminent speakers with us. I'll introduce the first speaker. The first speaker is Dr. Girdhari Lal. He's a senior scientist at the National Cancer Center for Cell Sciences, Pune. With a PhD, PhD in 2005 from ISC Bangalore, and later at Mount Sinai School of Medicine, where he was the assistant professor. He returned to India, and his lab since then has been working on understanding the cellular and molecular mechanisms of immune tolerance. He's the recipient of several fellowships and awards. At present, he's the vice president, West Zone of the Indian Immunology Society, and also the executive committee member of the Indian Oncology Society of India, Immuno-Oncology Society of India, and also of Society for Inflammation Research. Today, he's going to talk to us on mechanisms of immunopathogenesis in cancer. Pleasure to welcome Dr. Lal.
Thank you, Dr. Chiplankar. Let me have my slides on. Is it visible from your end?
Yes, we can.
Okay.
Yeah.
So let me start with Dr. Arora and Dr. Chiplankar for nice introduction, and Amit for putting this together. It's a wonderful meeting, and especially in COVID time, it makes more sense to go online. What actually I'm going to discuss today, what we have heard last 2 days, actually looking at different immune cells and how they function in different diseases, starting from infection, autoimmunity, and several other the diseases. Now, let me put this cartoon and then ask, especially from students, to select the best cartoon what they like, because we will discuss some of these cells, and almost all the cells what actually we know in immune system. You pick up any one of them, your favorite cartoon, and you will find all of them are interesting. Maybe the one you select will be the more interesting at your own way. Okay, so in immune system, when we talk about it's the different cell type, and most of the time laboratory, we take one of these cells and study and then define their function, phenotype, and then say that these are the cells, this is the cell which is important in one aspect or other. Now, when it comes to the cancer, there are a lot of immune cells. This kind of cartoon is very famous, and we know that we are always exposed to different carcinogens, but we all don't develop actually cancer. So the reason why we don't develop, because of alertness of our immune system. So we have normal healthy cells, and you get exposed to the radiation or any kind of mutagens, and then the cells become transformed. And once they transform, they are no longer same as what they are. And in that situation, our active immune cells recognize them, and using different mechanism, they try to remove those transformed cell, and that's how we are protected. But sometimes these tumors are smart. They actually modify themselves in a way that the immune cells which are here, which comes too close to the tumor, they actually no longer able to remove them. In fact, trouble is even more when the cells actually comes, they in the tumor microenvironment, they start supporting the tumor growth. There's a variety of ways tumor cells get support in order to grow themselves. And when they have the pathway is just not just one way, the tumor also build up something, some microenvironment in such a way, either in contact dependent manner or by producing some molecules, make these cells ablated from acting as a tumor anti-tumor cells. Now, if this is the strategy and this is said and done, how we can prove experimentally that yes, this exists, there are immune cells and they all are having checkpoint checks all the time so that there's no transformation, there's no existence of transformation cell. The best example I can give you this experiment, which is what is done here, one of the carcinogen, methylcholanthrin. What is done here is you take group of mice, give methylcholanthrin. There are a lot of mice which will not develop disease, and there are some mice which develop disease. So what you do is that after 200 days after the exposing to this drug, you select those mice who does not have disease. So now you have mice which is free of any tumor or visible tumor, and take these mice. Now these mice are exposed to the carcinogen, but they don't have a tumor. Now what you do is ablate the immune system. How do we do that? You give control antibody or antibody to different immune molecules. It can be CD4 T cell depletion, CD8, or cytokine neutralization. And when you do that, what you will find that the cells, the molecules, if you ablate the immune system, now the mice which were not having any tumor up to 200 days, now they start showing symptoms. So if what you saw here, the cell or molecules which were controlling the tumor growth, when you ablate them, now tumor grows drastically. You can go ahead and look for those different molecules and look at the tumor, what cell proliferating. So what it suggests that you have actually, when we are exposed, these immune cells are keeping us healthy by removing those transformed cells. All right, very good. And with the other example you can take, there are several knockout and transgenic mice where you have deficiency of some of these molecules, or the mice which do not have these mice develop spontaneous tumor, one or other type, depending on the nature of molecules. What it suggests that in any of these molecules which are there, they do contribute in the tumor development, in protecting us from the tumor. And when the moment you have there is defect or deficiency in either cell type or the molecules, then you will end up with the disease in terms of the tumor. All right, very good. And when we go to the laboratory to study the tumor, what we see, we take the tumor and then we start staining depending on the lab, depending on the choice of your objective. You stain for CD4, CD8, FOXP3, or macrophage or neutrophil, whatever you are interested in. And you stain for one and then do all those things and then say that, oh, this cell is very important, the other cell is very important. But actually, if you look in the context, there are a lot of cell types. And all these cell types work together in a positive way or in negative way. They talk to each other. And what is the overall output of all these cells in the microenvironment is going to decide what is going to be your cell effect in terms of tumor development. Okay, so whenever we have to study, I think we need to take context of the whole microenvironment and how it is going to support the tumor growth. Yes, there will be contribution of one cell type, but the other cells which are there, which we generally ignore, they also participate in one way or other way, in one way or other. This is another interesting slide. where you see the different cell types. What you see here, there are a lot of cells are infiltrating. It's not we're talking about just one cell type, CD4, CD8, macrophage, or whatever we name. And there is a heterogeneity in each one of these cell types. There is intercellular interaction and there is a molecular interaction within the cells to perform a specific function. So when you have a complex study like this, or a complex working environment within the tumor, then having everything from the petri dish and then say take one cell and say this cells is now transformed into this and that, does it really transform it? Probably not very good, because you require a complex environment. And this in complex environment, how these cell interact, whom they talk to, how they talk to, I think we need to understand that. So I will discuss some of these, how actually these are important and why we need to study them. That's another famous cartoon, you might have seen it. So in terms of when we say immunity, that means you should have antigen and you should have immune cell and you should have the generation of different immune cell types. So now let's imagine there's a tumor here, and now tumor cells are dying, or one way or there is damage, and then they are shedding the tumor antigen. Then the antigen presenting cell comes in picture. They grab these antigens, mobilize to secondary lymphoid organ, the closest one, that's lymph node, draining lymph node, where now comes picture of T cell and B cell. Now you have T cell activation, and then these T cells start expressing some cytochemokines, which direct them to go back into the tissue through the blood vessels or lymphatic vessels to the place where tumor originated. So now you see the cycle that antigen originate from one place, goes back to lymphoid organ, cells mobilize back in the form of trained immunity, in terms of trained against those molecules, the target, and now these cells comes in. Now this looks very simple, but if you look carefully, this actually require a lot of cellular cognition, the cellular interaction, as well as molecular interaction. And each one of these steps, depending on where you are looking at or where your eyes is focusing on, is a very important step. Now I'll show you after some in the following slides that there will be molecules which might be altering at this place or requiring at this place. And each one of them, you can target them as an anti-tumor therapy, depending on the nature and the tissue. Now, is the antigen which is you're targeting, is this specific to a specific tumor type? That's the best example again, here is given. So I showed you MCA, methylcholine. You give it to one mice, there will be tumor development. And you have another mice given the same tumor. Take it, mix it, and inject into the tumor, which then you take these mice, and then the mice will develop tumor. The one which has, so now what is happening? There is same antigen, two different mice, but if you take the antigen from this one and inject into this protect here, but it doesn't protect here. What it means? It means that if a carcinogen can generate n number of antigen, and it can be antigen different in different place in different organism. Now, nature of the antigen is the one which is going to protect. If the mice was the tumor is generated in this, if this is transferred in the mice, in other mice, which may be having different mutagenic characteristic and mutagenic antigen generated, it won't protect. Whereas there are examples where viruses, we know very small genetic composition, they carry very limited number of proteins. Now there are homologous, there are the virus which has closer similarity, and the one which don't have closer similarity. That's the best example here I can show you. So here SV40, you have SV40 just like same antigen, same carcinogen. Here two different, same virus in two different mice. You take these mice, inject into the separate mice, and then it doesn't matter from where actually you are giving, the mice will be protected. Whereas if you give this from another mice, now which is different type of virus, then a tumor will form. So it's not that it's the same virus which can protect. What actually you want to say here is that there is antigen specificity coming from the nature, the type of introduction of the carcinogen. It may introduce the antigen type. Now, what is important question in immunology, especially in tumor immunology? Can immune manipulation cause the regression of established human cancer? That's one of the major challenges, that you already have tumor. Now, how do we manipulate the immune system so the patient can actually get the benefit? of the developing immunotherapy, developed immunotherapy, or whatever way you want to manipulate. What are the antigen involved in immune recognition? And can anti-tumor T cell be generated in patient by immunization with the cancer antigen? If it is, what is fate going to be? What mechanism limit the cancer regression despite of in vivo generation of anti-tumor T cells? So that's another interesting area. So if you look in the patient, you do have T cells against the tumor antigen, but they are not functional. They are not functional, or they don't function at the place where they're supposed to be. I think that's a big question, and that's interesting question, and how to make them functional to the place where they are needed, not in the place in the circulation or in the circulation. All right, let's talk about something on antigen. So when you have tumor antigen, like I said, it's the tumor and the cell, the normal cells, they share a lot of features. There are certain molecules, it can be post-transcriptional modification in some antigen, post-translational, transcriptional, the alternative splicing, or there are several ways actually the antigen can be modified or mutated, and they are they can work as a different epitope, and then immune system recognize in those antigens. And also sometimes the molecules express on the tumor cells which actually protect them. So that's also interesting molecule. So not only just the antigen which is recognized by the immune cell because of alteration in the epitope, also the molecule which is expressed which can inhibit the immune response and then help in the tumor to survive better in the face of immune response in the body. Now, this is again a complex one. What I wanted to show here in microenvironment, what we're talking about is not so simple. It's not so easy. It requires the cells of different cell type origin, lymphatic origin. So you have blood vessels, immune cell, obviously, the B cell, myeloid cell. I think we heard all these cell types in the last 2 days, and all of them are here. So now, if you have all of these, which one is important? How we can make use of this to understand which is going to help the patient in terms of having anti-tumor immunity. Now it's much more complex. Okay. And when we talk about tumor involvement, depending on the tumor you're talking about is going to be different. You can take glioblastoma, now microenvironment is different. Skin, renal carcinoma, lung adenocarcinoma. So now you have a component which is totally different. Your immune cells are circulating, coming from the same location. Now it's challenging whom to talk and what kind of the tissue, what kind of therapy or target you should choose. Because if you just rely on immune cell to do everything, probably we are ignoring a lot of the fact that this microenvironment at different places are different. Something is different. So what makes it different? How immune cell responds in different conditions? I think that's very interesting area to explore. Very good. So now let's zoom on to tumor of different types. And when you do zoom on to the tumor type and go inside the tissue and look for the what is immune cell there, in broadly you can characterize into 3 different characteristics. One, you will find tumor with immune cells infiltrating almost everywhere, right? So you have tumor, you have immune cells, and you find a tumor where immune cells are localized in a specific place. They are excluded, but they're not inside the zone where they're supposed to be. So you have immune cell migrating, but not in the place where they're supposed to be to get rid of the tumor or kill the tumor cell. And there are tumor also exist where there's no immune cell. So now if you have this type of tumor, now if you think that you're going to use immunotherapy, which work in one case, cell apply everywhere, I think then you're taking a wrong decision. So depending on how the microenvironment and why immune cells in this place, despite of having a lot of immune cell, why they are not working. The cells are here, but then why they are not going? And the cells are not at all coming here. So if you have this type of tumor, now you can think of broadly how to manipulate the microenvironment or immune cells so that they can go at the right place and the right function with the right function so that you can have the anti-tumor immunity. Let's talk about this. So let's talk about a place where like this. So you have immune desert or there's an immune cell. What is missing here? What you can do to promote the immunity in a better way. So what you can do is you can actually first have antigen to be released from here in a way that immune cell come attracted to it. So that once they come in, they should be able to process and take up the antigen. And then once they take the antigen, they should be able to direct the immune response or develop the immune response. Now, how to do it? What are the things we need to think of? I think that's where the question mark is, that how we use different strategy to manipulate this type of microenvironment. Very good. Now you have another place, other type of tumor where you have immune cells around everywhere, but not in the tumor. So you don't have immune cell in this place. Now, if you don't have immune cells, tumor are not going to lyse just by looking at immune cells. So now the question is, how do we mobilize these cells here? So so that the immune cell recruited in the tumor and cross these stromal barriers and go inside the tumor mass. So you need to develop a strategy in order to mobilize them. So chemokine-based, cell adhesion-based strategy will work, where you need to redirect these cells to the right location. Now you have a tumor immune cells, but they are not functioning. If they are not functioning, we need to understand why they are not functioning. Is there a molecular defect? Is there a cellular composition defect? How do we engage these cells in tumor microenvironment so that now they start functioning? So these are the questions we need to look into it when we're designing a strategy for the immunotherapy. Now I will show a more complicated picture. What I want you to focus, what I explained to you, you have antigen, you have APCs, taking up antigen, going immune process, activating T cell, mobilizing through blood vessel, lymphatic vessel, coming back here. All these components, there are several molecules involved. Now, all these molecules which are in green, they actually contribute positively in the developing the anti-tumor immunity. The molecules which are in red, they act as inhibitory factor depending on where you're talking about. Now, if you're talking about lymph node, all these components positively or negatively in a processing and presentation or antigen setting, or the molecule need to be activated, mobilization of these molecules. So now you can understand the complexity of the disease. So that different level, different molecules, it can be chemokine, adhesion factor, or other cellular cytokine or other factors required in order to have the proper function. Very good. Let's now look at how actually in general condition, other ways the tumor develops. So now you have a normal skin, you have some kind of infection, a persistent infection for a long time, and infection goes away, but it leaves something, sometimes the infection keep on going. And in that case, you have a lot of reactive oxygen or the obnoxious molecules produce all the time, and that can cause DNA damage, or the molecules which can damage the cell and tissue around. And when you have this type, there's a transformation of the cells, and that can also lead to the tumor development. So now there are different ways tumor can develop, different ways that we can have checkpoints. Now you have tumor, let's go more inside the tumor and see what is going on in terms of molecules and cells. So you have a cell, which is tumor cells. Now they're expressing antigen, and you're also having CD4, CD8 T cells. They actually produce or invite somehow the molecule, when it comes as a regulatory cells, MDSC, myeloid-derived suppressor cells, they produce molecules which inhibit the function of effector cells. We heard from last 2 days, they are effector cell, or a cell which can actually have anti-tumor effect or the inflammatory nature. You have other cell type like macrophages. We'll discuss this molecule, this M2 and M1 macrophages. There are molecules which inhibit the function of this, and that's where the checkpoint inhibitor comes in picture. I think you will have a nice lecture in the evening. And there are molecules which actually blocks a lot of things. So there is a complex biology where cellular and molecular interaction happens, where the effector function or inflammatory function of different immune cells are blocked. There are cell types which actually differentiate in the microenvironment, and they produce a suppressive microenvironment, and that's where tumor enjoys. Now, any of these molecules, if you look in literature, you can target them, and you will find them that they actually somehow contribute in, if you inhibit or block, they contribute in protecting from the tumor. All right, so when you have tumor, it's not just tumor develop in microenvironment. They are more, sometimes they become aggressive and they decide to go away from the place where they originated. So now question is how they decide where to go, because they definitely has to travel through circulation. And when they have to travel through circulation, they are allowed to go everywhere, almost wherever the blood circulation is. But that doesn't happen. We don't have metastasis, the word is used, where the cells move from one place to the other location. And in this metastasis happen in a specific organ in a specific place, depending on the nature of tumor. So what dictates? What dictates that which organ now they are destined to? And imagine that in most of the time in the blood, when you look at it, you will find the cells are actually traveling to the highly inflamed microenvironment. So moment cells decide to go away, they can be killed on the way. So they must design a strategy to protect themselves. And how do they do it? So let's look at in this slide. So you have a tumor cells, you have vascularization to begin with. So you have highly vascularized place. Now in the vascularized place, some cells, you have to dissolve the wall of blood vessels. So cancer cells can go in. Now once they go in, they are actually now in highly inflamed microenvironment in circulation. So what they do actually, the moment they land in the vessels, they actually invite a lot of suppressor cell. It can be MDSC, platelets, other cells. So all of them form an emboli, and they surround in a way that now they are no longer alone or free floating cancer cell. Now when they go in, they are carrying now immunosuppressive microenvironment around them. So when they go, obviously because you're growing in a emboli or in bulk, they get stuck. They go to stuck wherever there is a fine capillary. Where are the fine capillaries? We know the lung, we know the kidney, we know the liver. A lot of fine capillaries there, and metastasis most of the time happen. They get stuck. Not only that, they also directed by the chemokine and chemokine receptor and cell adhesion molecule in a specific tissue. So once they go, they now, because they have reached to destination and protected and guarded by the cell type like MDSCs and other inhibitory cells, now they develop secondary tumor, and that's the metastasis. All right, very good. We're talking about macrophage, M1 and M2 macrophage. I think Dr. Chen also mentioned about it. And the M1 and M2 molecules depend macrophages, and sometimes when you have interferon gamma, LPS, classically, if you take monocyte and activate, that's called classical macrophage. And there are other other way you can activate macrophage, that's called alternative macrophage, where you use IL-4 and other molecules. So if you look at these molecules belong to like what we know at Th1 or Th2 side. So when you have to, don't get confused with M1 and M2, just look for what you define as Th1, the molecule which is producing. That's look like that's the way we are going to name them. Now, what they produce actually, what they produce here, you can look at these cytokines. These are now suppressive cytokines. So that's how M2 is labeled as inhibitory in nature, and M1 is inflammatory in nature. And originally it was M1 and M2 was discovered based on the amino acid, what they were metabolizing. So if you look at, people call it as bulb C57 mice are mostly M1 type, or they generate M1 immunity. Bulb C has M2. And if you look at, there is a gene which is defective, which is arginine transporter in macrophages. Because of this defect, now M1 phenotype is dominant. And that's how these mice, and that's how the susceptibility to different infection in these two different strains of mice is different. All right, some of the ignorant cell, that is eosinophils. Eosinophils, now we know, now things are coming that because they carry molecules and granule which can kill the tumor cells or activate other cells, now eosinophils are also considering getting more attention in terms of its anti-tumor activity. And in fact, look at in the different tumor where eosinophils are more, aggressiveness of tumor is reduced. We need to learn more about these cells, but there is actually now more attention on these eosinophils and their role in anti-tumor immunity. Now coming back to again this whole circle, now I was telling you different molecule which has green and red. Now we're talking about molecule which is antibody. So now these molecules which are here, which is required in order for the APC and T cell interaction, you see these molecules are used. I think this will be discussed in more detail in the afternoon. But I wanted to point out at each step, you can think of those molecules to block this, and you can think of those as an anti-tumor agent. All right. Now let's closer look at this in terms of what I was telling you in terms of where the cells mature. And when we're designing this strategy, we need to keep in mind that how they are going to work and what is their requirement in the basic place. So for the student, just look into this. The spleen is not the mixture of T and B cells wherever you feel like you can put them. They have very well organized structure, the follicle, T cell zone, and then the red pulp. And they are surrounded by specific cell type, and these cell types like medullary macrophages, they are surrounding all the time. And what it means to immunologists that it's not the T cell and B cell wherever they feel like they can go and they can do their job. They have to go in a very systematic, very specific manner. All right. So when the cells doesn't fall in the right place, or if you alter any of these things by any ways, either by using antibody or the chemicals, actually you alter the whole immunity. So let's not discuss that part today. Let's move on to the more structure. Like I was telling you, there's requirement of lymph node, and you know the cell's lymph node structure. There's a very well organized structure. You have a defined area where T cell exists, naive T cell, and defined way that immune cell enters, and then they interact with the naive cells in germinal center. The only naive cells go in a different ways. If you have activated cell, they can directly land here. So the way they go, the way they interact, that makes actually very important thing. So if antigen comes from here, it has to travel all the way this, so immunity can develop. Now imagine that you develop a molecule and it's very immunogenic in petri dish, because you actually have cells in petri dish, anything can contact, and a nanoparticle, especially nanoparticle based immunotherapy. Now, these molecules cannot travel in this way directly because of the size and the nature of the molecule. If they don't come here, what you see in petri dish never be translated in terms of immunity. So they have to travel in a specific organized manner, a specific location, and there are immune cell which does a specific job. And if that is not done, then immunity is not going to work out. I know that this type of structure, this type of interaction is shown very well, but I'll take one example from here to justify that why it is important. Like I was mentioning to you, there are certain situations where the antigen cannot go through because of size. What people has tried directly into inject the location where the interaction of immune cell happen, and in that case, you can mount the immune response. So the location and the structure and the movement of molecule in a specific location is very important. If it's not done correct way, immunity in vivo, what you going to expecting in vitro, you will never achieve in vivo. And that's the major reason most of the time we fail. Now, what is the structure in terms of immunotherapy? So now let's take example here. We know checkpoint inhibitors, PD-1, PD-L1. What is done here is that you take mouse and then you give the MC31 adenocarcinoma cell line in B6 mice. What you do, just remove the lymph node and then ask question, if I give checkpoint inhibitor without lymph node, can I develop immunity or can we develop anti-tumor immunity? So here, no treatment, mock treatment. Here what you have done, if you give the PDL1, the tumor is regressed, remove the tumor, now you don't have, immunity is lost. Similarly, survival of the mouse is altered. So what is suggesting is that you require lymph node in even any of these molecules to function, because by the nature of the way immune cell interact and the location they interact, that plays very important role. All right, now let's discuss something on different cell types. Gamma delta, I don't think we have time to discuss, but what I wanted to point that there are cell types which are minor in nature, but they can do a lot of things in terms of anti-tumor immunity. And there are specific gamma delta T cells. People are trying the clinical trial, either modifying the receptor or altering in vitro, infusing back into the patient and see the tumor anti-tumor property. Other cell type is NK cell, which I will discuss little more on this. So you have the patient, you take the PBMC, purify NK cell, in vitro activate or modify, inject back into the patient, and then people try it that way. Or you purify this, engineer them into using recombinant technology so that they recognize the specific target, and then you can do that way. Or you can modify different activating and inhibitory receptor on NK cell. Let's look at more detail, and that's how this slide is showing that a lot of these NK cell trial has been done, because thinking that NK cell, when you infuse or when you modify in vitro, they actually get more cytotoxic property. But most of these actually failed when they did a real clinical trial. The reason why they fail is that in petri dish you generate a lot of these things and they all are functional, but in reality, most of them, when they do not land in the right place in the right time microenvironment, they're not going to work. So that's where we're interested in the lab to find out why NK cell actually fail. And then this is simple experiment is done. So you take B6 mice, give the tumor cells, and the tumor develop, and then the tumor is growing. and just come back in within 5 days and then ask, is there NK cell in the tumor? So you can find there are a lot of tumor actually NK cell in the tumor even so early and the late in the tumor microenvironment. So now if you have NK cell, why still tumor progress? What is the problem with these NK cell? Why they are not able to kill? And if you take these NK cells in vitro from the spleen, they will able to lyse the cells. So now what is the problem? What is the phenotype of different NK cell? So based on the two molecules, CD27 and CD11B. If you look at the expression of these molecules, CD27 and CD11B, the molecules which are double positive, the CD27 and CD11B, both double positive, they're highly cytotoxic. And the molecule which has very low expression of this CD27 low and CD11B, they are less toxic. So you have the molecule, the NK cell which are expressing these molecules, highly cytotoxic. Then we ask, okay, let's look into the tumor, what kind of NK cell you have. So in the experiment, Saurav Paul from the lab, actually, he looked at NK cell in the tumor. What you find is CD20C, CD11B positive. In tumor, actually, you have a lot of these cells which are double positive, which I showed you that they are actually cytotoxic. Now they are not, if they are highly cytotoxic, why they are not functioning? So I think I'm not going to discuss more detail on this data. So what I can tell you, what happened in those cells. So you have two NK cell, the way NK cell function, and they are different from CD8 T cell, that they look for MSC class 1 absence. So if a cells which don't have the MSC class 1, they recognize through inhibitory or activating receptor, and they find that if the MSC class 1 is missing, the inhibitory receptors, the function of inhibitory receptor is gone, and activating receptor get overdosed, and then the NK cell kills them. So lack of MSC class 1 is going to be detected by NK cell. And what are those inhibitory molecules or activating molecules? Here I'm showing you the activating receptor. There are different types of activating receptor. And if you look into the tumor, in fact, even though their double positive cells are higher, they actually lack less, or they have less activating receptor in the tumor compared to the outside the tumor. Now, if you look at inhibitory receptor, they are actually more dominating in the tumor and outside the tumor in the same mice. So you have a different composition of this inhibitory and activating receptor. And we know that that's one way of working. I'm not going to discuss detail, but I wanted to point out the way they work.
Sorry,
so now,
sorry, Girdhari, you have to
yes,
wind up in 5 minutes.
Yes, I will do. I'm very close. So here you have the NK cell, which also work by producing cytokine, which is interferon gamma. And interferon gamma, we know that help other cell type. And then you have CD8 T cells. So then we also looked at how they work. And what we find is here, you take this NK cell, you can take these mice and deplete NK cell. And then you see that depletion of NK cell actually have more tumor cells, tumor development. What I wanted to show you that the way NK cell was working is also one way, indirectly, by actually promoting more T-beta positive cells or Th1 cell response in tumor. You can look into this Spleen, you can look into the by flow. So essentially, the NK cell was contributing in immunity. Another cell type is NKT cell, another subpopulation which actually shares the T cell receptor and NK cell receptor. Now, in this one, we know that NK cells are actually less in the tumor microenvironment as the time proceeds. So we thought, okay, let's activate. There are a lot of NKT cells in this. And if you activate with one alpha-galactosyl-ceramide, which is the one lipid molecule, and then if you do that, the tumor actually gets regressed. And if you look at the NK cells, actually, you can look at the alpha-galactosyl-ceramide positive NK cell in the spleen or tumor. And in that case, you will also see that NKT cell, if you deplete, or activation is gone if you deplete the NK cell. There are other antibody which can specifically deplete NKT cell. Let's ignore that. So in somehow NK and NKT cells are contributing in Th1 response. And also, if you look at M1 and M2 macrophage, we discuss about M1, like you can mark by INOS or by CD26. So when you have galactosyl-ceramide treatment, not only work on NKT cell actually promote the Th1 response predominantly, and that's how it works. So to summarize this part, what I said, NK cell and NKT cells, they directly work through either tumor directly by lysing it or by producing some cytokines and then helping other cells in order to generate the anti-tumor immunity. In nutshell, what I'll say that immune cells comes in different flavors. So you depending on what you want to see as the good hand or the bad hand or the arm, the right arm in the right hand in the right place is the one which is going to help you in the immunity. Thank you. I will take the question from here.
Thank you, Girdhari, for finishing in time and for a very elusive presentation and projecting both the sides of the anti-tumor immune system, immune surveillance, as well as how immune system contributes to pathogenesis. There are questions, but I think we'll take the questions after the second talk by Gauri Shankar. We'll take it together.
Perfect.
Thank you.
Ma'am, your camera came on. Your camera is on now. We can see.
Yes.
Very nice.
Okay. So let's now invite the second speaker, Dr. Gauri Shankar Shah. He is the professor and chairman of molecular medicine at Bose Institute of Kolkata and also the executive chairman, Center for Translational Animal Research. His research work focuses on cancer biology, tumor immunology, and understanding the molecular mechanisms. His recent work is on immune evasion and immunotherapy of cancer. He is the fellow of the National Academy of Science, and fellow of the Nanyang Academy at Singapore, and also the West Bengal Academy of Science and Technology. He is going to talk to us today on cancer vaccines, past, present, and future. Gauri. Is he there? I can see him, but...
Yeah, he had a lot of network problem, but he has come back. Yeah.
He has come back. Yeah, he will be joining.
Good afternoon. Can you listen to me, Dr. Gauri Shankar?
He's not audible.
Hello?
Gauri is not audible.
He had a lot of problem in connecting, but ultimately he was able to connect.
Oh, I see.
There is some problem with his network.
In the meantime, then, should we take the question? There are some questions.
Sure.
I think he's not able to locate how to unmute.
He's unmuted.
Unmute?
No.
No.
I think problem is.
Ma'am, in the meanwhile, Girdhari can take up questions. You can.
Yeah. There are some questions. Girdhari, I think you addressed it, but there are two questions from Ms. Arya. And one is on how effective nanoparticle-based immunotherapy would be in drug delivery system. And the second question again is a take forward from that, where the nanoparticles with checkpoint blockade siRNA and combination nanoparticles with small molecule checkpoint inhibitors, would similar strategies work? What might be the plus and negative of this combination therapy?
Okay, so let's talk about first nanoparticle and its efficacy. So now the first thing what you will do in the lab is that you find out a nanoparticle which is blocking either the checkpoint molecules. You will do in vitro and then find out that yes, they are doing their job. But when you come to the in vivo, what I said to you that the nanoparticle has to go to the right place. They need to block the interaction in the lymph node, not in the circulation, because that's where not going to help. So you pump nanoparticles and it goes in circulation is not going to work. So it has to cross those barrier, the small microvesicles in the lymph node or in the spleen, so that they should actually alter this interaction. If you able to succeed, and if your size of the molecules, nature and hydrophobic, hydrophilic, or any other ways that it can do it, I think that will have chances to success better. Then
there is the second question by Krishna Chaitanya, and it's on how anti-inflammatory agents work by shifting M1 macrophages to M2.
Right. So when we say how anti-inflammatory molecules work, so like I said, it's just not one cell or one molecules. It's the microenvironment where the cells are going to work is important. So if let's say if I say anti-inflammatory molecule, just one molecule, now it has function at different cell types, not just on M1. Even if it is M1, M1 is going to contact a lot of other cells either directly or indirectly through producing some other cytokines. So how it is going to work is depending on how efficiently it is going to alter the cells in the microenvironment. So if we can achieve that, I think it's going to be more interesting than just what we see that it alters in vitro or alters in the petri dish.
The next question is how anti-inflammatory agents effects on pro-inflammatory and anti-inflammatory. Oh, sorry, we took that question, right?
Yeah.
No? Yeah. So then another one is, sir, what is the specific pathway to be which region has to be chosen for better expression? I think it's a very vague question. It's by Michael Burairaj.
Okay, which region to be chosen? I think that if his question is that if you're looking at histology of the tumor and then you're trying to figure out what to target and where to target,
Yes,
it's the now nature of the tissue microenvironment and what is missing. We need to figure out is the chemokine what is required or cell adhesion molecule is required in order for immune cell to localize. If that is there, that can be altered or that can be promoted depending on the what we are looking at. If we can manipulate that, I think the immunity will be much better. And now mobilizing the cells, let's say if I have a cell which is highly cytotoxic cell, but it doesn't go there. So why it doesn't go to the place? That means it doesn't have right chemokine receptor on them or cell adhesion molecule which guide them. If so, we need to have cell type which should also have this molecule on their surface so that we can achieve the mobilization of this cell to the right place.
Dr. Sunil, there are two more questions, but if Gauri Shankar is ready, should we take his talk? Hello? Dr. Arora?
Yeah, you can take up the questions. He's still trying.
Okay. Then another question, the person has not written name. What is the role of circulating tumor cells?
Role of circulating tumor cell. So is it mean that he's talking about lymphoma?
No, I think it's in terms of diagnostic because there's a lot of emphasis on circulating tumor cells.
Okay, so in terms of circulating tumor cells, it might be kind of like metastatic level that from benign to actually is going to metastatic phase. So that's alarm that what tumor cell or what cancer type you are referring to. Depending on that, we can actually look into the nature of the depth of the what you should see in the clinic.
Yeah, because today nowadays a lot of emphasis on circulating tumor cells and their importance in cancer.
Right. Yes.
There's another question from Ms. Rajeshwari Farida on what's the mechanism of cancer autoimmunity?
Cancer autoimmunity. Okay, so now that's very that's very good example. I'll tell you that in melanoma. In melanoma, you know tyrosinase. Tyrosinase mostly expressed very low level, but when there's a melanoma, this molecule expressed very high level. Now, this is a molecule which is self-antigen. Now you have a lot of immune response as well. If you look at a lot of melanoma, you'll find antibody against this molecule. So if any of these molecules which are highly expressed, aberrantly high expressed, which are self-antigen, not mutated, but they are actually expressed. So the advantage sometimes in diagnostic it helps because if some of these molecule can be used as a diagnostic molecule, and in terms of autoimmunity in long run, it may have, there are certain cases where they do develop autoimmunity. So some of these molecules do altered, but the nature, because we have the anti-inflammatory microenvironment, it doesn't develop that strong.
I think the lecture has stirred a lot of interest. There are more questions coming in. Then another one from Dr. Rakesh Sharda. What is antigen response or sensitization therapy?
Okay, so sensitization therapy or antigen response, obviously, like I showed in the slide, that if you have a tumor where there is a desert, you require the immune cell to first get activated, and then in antigen sensitization does help, because you in the circulation you build up those cell type which are specific to the tumor antigen, and now if they are guided by the specific chemokine, you see that these cells do mobilize in the tumor, and they will have anti-tumor activity.
There's another one. Oh, the questions are coming in, pouring in. There's another one from Magapu Solomon Sudhakar.
If Dr. Gauri can, ma'am, can you
Gauri
try?
Shankar ready?
Dr. Gauri Shankar?
Should we take his talk now?
Hello?
His audio is not I mean, we're not
cannot
able
be
to
heard.
hear him. Is he using Mac?
Yeah, I think he has a lot of problem.
Dr. Gauri, are you using Mac computer? So probably you need to go to
He's using
setting.
Mac. Dr. Vidari, and he's using
Yes.
Mac. That's why I
So
think we are
he might have to go to setting. He might have to go to setting, and in the privacy, there is a option where he has to click Zoom. If he click the Zoom and select Zoom also, Zoom, then probably it will work. Microphone and Zoom in the privacy setting.
Yeah, you're
I
right.
see sometimes this problem with some Zoom link.
Okay, in the meantime, should we continue with the questions?
Sure.
Then there's a question from Magapu Solomon Sodhakar, that is, sir, how much success is gained through precision medicine in treating GI cancers?
Okay, so very good question. I just missed, actually. I had some slide, but because of time limit, we couldn't able to do it. So now precision medicine, that's very, very interesting area. Now, what we know, how we learn. So first thing we need to find out what is the antigen which are unique in any of GI tract infection, the tumor like colon cancer, or how do we find out that which cell type, which particular antigen? And when we find out this, which kind of immune response we are interested in? Is it humoral response, which probably doesn't work well, especially in GI tract? So we need to depend on cellular response. Now, which cell type we should actually look for? It is the modified T cell, what we call it now, genetically modified T cell or CAR T cell, chimeric antigen specific cells. Once you have antigen, specific target are frozen or designed, then you can actually generate those T cells specific to those colon cancer specific cell, and then you can have the immunity using those cells, modified cells, as a therapy.
There's another question from Ms. Nima Vora. Can glycopeptide be a possible strategy?
Can glycopeptide be a, right? So there are right. So there are, like I showed in one of the slide, that there is a post-translational modification in some of the molecules, and when these molecules are altered, they are immune cells which is recognized. Now, when these molecules are recognized, they can be act as a target. But now question is, who recognize and how it works? So the most of the time, what happened with glycopeptide, you mostly generate the humoral response, not a much better T cell response. So in terms of humoral response, you know that it's very good that if you have a humoral response or antibody which can generate ADCC or antibody dependent cytotoxicity, then it might work. And in T cell response, obviously you have a challenge to do that. So depending on the what and how it's going to work, it may have effect on it. Yeah.
So that's all from regarding questions.
So, ma'am, you can open it to the panelists for the time being.
Okay.
I don't know.
Okay, you're still trying to get the connection. Okay, so questions from the panelists? If not, Girdhari, I would like so whether we can have some discussion on neoantigens.
Okay.
I thought that would be something of interest, you know. Oh, there is one question which has come again. Then we
Okay.
take it. There's a question from the participant, from Michael Durairaj again. The mechanism by which there is variation between mechanisms of benign and malignant cases.
Okay, so now we need to see that when we call it as benign and when we call it as malignant cells. So in case of benign, you have something which is just stay where they are and doesn't have much impact in terms of going out of their territory. So now why something goes out of territory and what are the requirement for in order for it to move? Obviously, you have more mobility when you have roadblock is removed, and that means you build up more road, and that means vascularization. If you have more vascularization somehow induced, and also the connectivity, the vehicle and transport is set up, you mobilize much better way. So same thing happened here in the tumor. So in benign tumor, you don't have that much the requirement for going outside, and also the requirement in terms of setting up the requirements so that they can go out. So the mechanism, what I said, is again cellular and molecular interaction. The chemokine receptors, what they require to mobilize immune cell in, and also if the tumor cells out, or tumor even immune cells to go out to generate the immunity. So I think there is a different requirement of different mechanism in terms of requirement of different molecules in the benign as well as malignant cancer.
We a little bit more we can discuss, since we have time, on the
Yeah.
immune evasion strategies, which
Right.
you showed in your presentation. So
Yeah.
whatever you do or manipulate the immune system, the tumor finds a way to evade being
Right.
recognized.
Right.
And that makes it very challenging for the immune system, I think.
Right.
So like downregulation of MHC1 or
That's
so whatever you do to manipulate the immune system, there is a counter action by the tumor.
right. So
And
that's very
that-
interesting area. I think I missed those some of those information due to lack of time, but that's very interesting area. So the where is different way. Now I'll discuss some of those things, like
yeah,
the Dr. Chaplunker mentioned MSC class 1 down regulation. So when you don't have MSC class 2 on the tumor cells, then you actually getting off from CD8 T cell response. But at the same time, you also invite NK cells. They actually look for absence of MSC class 1. So now here is the strategy. Now you don't have one arm, the other arm is trying to again act on it, but the tumors are also smart. They have other strategy that the metabolic requirement in the tumor microenvironment, they alter in such a way that even the cells which doesn't look for even absence, like look for the absence of MSC class 1, they are now ablated. The example you can see the adenosine or something nucleotide metabolism, which is there in microenvironment. You have other cell adhesion or inhibitory molecules on the surface expressed, and then the molecule which actually promotes the inflammatory response and generates more cytokine, which generates anti-inflammatory molecules. So somehow this immunoediting, what they do in tumor to generate more anti-inflammatory microenvironment, even cells come with different armament, they actually lose their function in the tumor microenvironment. And that's how probably tumor enjoys. In fact, they start helping the tumor in one way in the tumor microenvironment. Yes.
So, Girdhari, can I
Yes.
ask you something more? You
Sure.
mentioned there is a in the tumor sometimes there's a although you somebody else also asked, I want to know more about it. Like when there are no immune cells in the tumor, there is an immune desert. So you said that you have to trigger something to get the immune cells into the tumor. So you mentioned that you have to put some antigen. Do you think besides tumor antigen, if tumor is not expressing antigen or neoantigens or whatever, so is there any other way that you activate the migration of immune cells to the tumor?
So that's very good question. You need to have right postal address for immune cell to go. If they're not having the right address where to go, you might have to tell them that this is the address and this is the way you go. So when we actually generate immune cells in the body, we also need to make sure that they are express the right chemokine receptor and cell adhesion molecule, and same time the deserted tumor should express chemokine so that they get the clue that this is the place I need to go. So we need to design a strategy where there will be activation of tumor where these chemokines are produced, and when they are produced, simultaneously the immune cells which are supposed to go there, they express right the cognate chemokine receptor. If they are expressing the same, then you will have success of mobilizing the immune cells.
Right. So that's very, hey,
Is
very
it?
nice. Good to see you. Yeah. Come on.
Yeah.
Now we can
Now
hear.
you can hear me?
Yes.
Yes,
Yes.
yes, both here and
Oh,
see also
thank
both.
God. Yeah, I'm now joining from my PC.
Okay, perfect.
Oh, okay.
Okay.
So, shall I
So
shall I
start?
start?
Yeah,
Gauri,
sure.
introduce you.
Yeah.
Your
Your
phenotype
phenotype
has
has
changed.
changed.
You look different. Okay, it's my pleasure to now welcome the second speaker, Dr. Gauri Shankar Saha. He's professor and chairman of molecular medicine at Bose Institute, Kolkata, and executive chairman, Center for Translational Animal Research. His research work focuses on understanding basics of cancer biology and tumor immunology and understanding the molecular mechanisms. His recent work is on immune evasion of tumors and immunotherapy of cancer. He's a fellow of the National Academy of Science, fellow of Nanyang Academy of at Singapore and West Bengal Academy of Science and Technology. His talk today is on cancer vaccines, past, present, and future. Welcome, Dr. Gauri.
Yeah. I'm sorry because I had lots of problem in logging from my MacBook. So now I switched to my PC. You can see is cancer vaccine. Uh, hello.
Hello,
Can
you're
you
audible.
hear? Okay.
Yes, you're.
In the cancer vaccine, past, present, and future. Actually, I wanted to talk about immunotherapy. So Sunil switched me to talk about the cancer vaccine. Uh, Let me introduce the cancer. Cancer is not an affliction of the modern world. The scientist has discovered the first known fossilized brain tumor belonging to Tyrannosaurus rex 72 million years ago. So cancer is not an effect of the modern science. It was also evidence millions, trillions of years ago. And the oldest description of cancer was discovered in Egypt, date back 300 BC. And the oldest available specimen for human cancer was found in the mummified skeleton. So as you can see, that cancer is a very old disease. So that's why we were interested in doing research in cancer. So why cancer? As you can see from this demograph, that with in the last 60 years, you can see that there is a lot of discoveries in medicinal system. That's why the mortality rate in different diseases decreased drastically. But till 2010, the change in mortality rate in cancer didn't change much. Only a few 5 to 7% patients they could survive from the cancer because of the discovery of some chemotherapy. But if you look back recently, after the discovery of the immunotherapy, the scenario has is changing. You can see that after the discovery of this immunotherapy, the percent of survival is increasing dramatically from 5% to 20% in last 10 years. So that's because of this immunotherapy. So so immunotherapy is a new class of cancer treatment considered by many to be the fourth modality of cancer treatment after the chemotherapy, radiotherapy, and surgery. And based on utilizing the patient's immune system to fight the cancer. And cancer vaccine falls under this category of treatment. So cancer vaccine is a part of the immunotherapy of cancer. So, what is immunotherapy? This is for the student who doesn't know properly what is the immunotherapy. So, it is something like that. When you see some tumor in your body by the mutational because of the mutation in our body because of the exposure to the different carcinogens. After certain time, when the tumor grows, after certain time, when it becomes nearly 8 to 10 millimeter in size, then we can recognize the cancer, and the patient, he goes to doctor. Doctor gives him the first degree of chemotherapy. The tumor shrinks, and the patient goes to holiday. After some time, it may be sometimes for 1 year, after 2 years, the tumor again come back. And by that time, some certain percentage of patients, they die. Those who are lucky, they respond to this tumor, and after 1 or 2 years, the tumor come back. Then the patient again go to the doctor. Doctor give him the second degree of chemotherapy, which is more harsh than the natural chemotherapy, first degree of chemotherapy. Again, the response, the tumor size shrink. But in a short span of time, again the tumor come back. So again, have to give the third degree of chemotherapy. In that case, most of the patients, they die. They can't tolerate this chemotherapy, or again the tumor come back. And at a certain time, the patient dies. That's why the mortality in cancer is so high, despite the discovery of number of chemotherapy. So the scientists think if we can start the immunotherapy either this point or at this point, we'll be able to really, get the better results, and the tumor will shrink, and the patient will survive. That's the mode of action of immunotherapy. So, but the concept of immunotherapy is not new. 60 years back, 70 years back, Peter Bryan and Frank, they first thought about they did the first scientific experimentation on the involvement of immune system in cancer, and they discovered the immune surveillance of tumor. For that, they got the Nobel Prize, but after that, their work was not very much recognized because there was no good tool to understand or to prove their hypothesis. In fact, the concept of immunotherapy is even older than the work of the Frank and Bryan. If you look at our Vedic literature in Charak Samhita, you will find the information about the immunotherapy. At that time, in 2500 to 400 BC, when in the Charak Samhita, there was a verse which says that at that time, the patient, our Vaidyas, they used to do the immunotherapy. What they used to do, they used to cut the tumor mass and let the blood from their siblings go into the tumor mass, and after sometimes, after a few days, they find that the tumor is shrinking. So actually the blood is carrying some immune cell from their siblings to the patient's the tumor, and this immune cells, they are killing the tumor. So that is the first evidence of the immunotherapy in literature. So vaccine, as you know, the one part of the immunotherapy is the vaccine. So what is vaccine? The vaccines have proven effective in suppressing different diseases because the vaccine was developed in 2,200 years ago. They have prevented some of the 20th century deadliest diseases and have helped several hundred or hundreds of million people. And cancer vaccines are the form of immunotherapy that can help and educate the immune system about what cancer cells look like, so that it can be recognized and eliminated by our immune system. So this is the cancer vaccine. So if you look at the historical first rate in 1777, note the surgeon of Duke Kent first inoculated cancer in one patient to himself. So he took a cancer tissue from a patient and inoculated it in himself. In 1808, Alliard, the physicians of the Louis 18, received an injection of breast cancer material from a cancer patient in his body. And in 1970s, the scientists have started producing the monoclonal antibody against the tumor so that they can use it as a cancer vaccine. This is the history of how this cancer vaccine developed or the immunotherapy developed. As you see, the PD-L1, the latest development of CAR T-cell, anti-PD-L1 and anti-PD-1 body, and different types of cancer vaccines were evolved from 2010 to for the last 10 years. It evolves very much. So cancer vaccines are something like to aim the cancer vaccines to stimulate the immune system to able to recognize cancer cells as an abnormal and destroy them. There are two major categories of this cancer vaccine. One is the preventive, or which is called the prophylactic, and the other is for the treatment or therapeutic. Each of the cancer vaccines work on the same basis. That means it can stimulate a distorted cell in our body or defective cell in our body and try to kill them or eliminate them. So this is the basic concept of all types of cancer vaccines, even the immunotherapy. So anti-tumor response, like prophylactic vaccines given to the people, those who are in high risk of developing cancer. So, they give them some boosting for the immunotherapy, the vaccine based on the antigen of the cancer. So if someone is at the high risk of breast cancer, so they give the antigen of the breast tumor cells that stimulates their immune through the immunomodulatory circuit, and that activates the Langerhans cells in the body. These cells then activate then they go to the immune compartment and activate the B cells to develop specific antibody to the cancer. And then the B cells are going to the clonal expansion so that they can produce a large amount of antibody. So secondary immune response, like the large number of T cells are activated, high titers of antibody are developed, and it continuously activates our dendritic cell in our body so that the tumor will not be able to grow in larger volume and eliminate as a secondary immune response. So this is the prophylactic vaccine, type of prophylactic vaccines. And so you can see this is the when the tumor is when the antigen is inoculated into the patient body, it activates the Langer cells, and these Langer cells then they migrate into the lymph node where they activate the plasma cells to develop yeah. So they are the B cells are produce the antibody, and there is a clonal expansion, and they produce high amount of antibody so that they can target cancer cells and kill them. And there are other types of cancer vaccines, which is called the therapeutic vaccine. What is happening? Therapeutic vaccines where the where the cancer produce when the antigen from the cancer admits of a tumor antigen into the patient so that the patient's immune cells are like Langerhans cells are activated and presented to to the T cells. These T cells activate B cells that can produce specific antibodies. And migration of the tumor sensitive T cells to the tumor site, and they inhibits the tumor growth. The problem with this, the prophylactic therapeutic vaccine is that during the tumor development, there is a lot of immunosuppression in the tumor microenvironment, and the establishment of tumor heterogeneity that leading to the tumor antigen expression and less number of antibody development. And the tumor cells, they don't sometimes recognize the T cells, and they are resistant to the immune destruction. So these two types of cancer vaccines are available. So there are different types. Out of these two prophylactics and therapeutic, there are a number of cancer vaccines that have been developed. One is the dendritic cell vaccine. Other one is the antigen vaccine, anti-idiotopic type vaccine, DNA vaccine, and tumor cell vaccine. Dendritic cell vaccines are the dendritic cells break the antigen of the cancer cell surface into the smaller pieces, and then this DC then act as the most wanted poster to the immune cell, displaying this antigen to the killer T cells, and this killer T cell, they attack this tumor. They sometimes, what is happening? Why it is doing that? There is some problem with the, yeah. And this dendritic cell then stimulate our immune system like T cells, and the T cells then activate the B cells to produce antibody against this tumor. So the people, this are then exposed, the CD from the cancer patient are isolated, and they are exposed to the antigen from the patient cancer cells. And this combination of the DC with the antigen is then injected into the patient, and the dendritic cells work as a program and activate the T cells, so that the T cell can activate B cell to develop antibody. The Provigen is one of the example of successful dendritic cell vaccine. And there are some antigen vaccines. The tumor-specific antigen protein displays on the tumor cells stimulate our immune system. So by injecting large amount of this antigen from the cancerous area, the immune system will produce increased amount of antibody or activate the cytotoxic T cells, so that these cells can kill the cancer and stop the tumor growth. Multiple antigen can be used for this to develop the cancer vaccine against the antigen. The examples of such antigen vaccines are tyrosinase GP100 or MART3 peptide or NY-ESO, which is being tissue cancer. See, and there are other types of vaccine which is called anti-idiotype vaccine. Some antibodies called idiotype antibody can act as an antigen for this cancer, and they trigger the immune response. These are primarily used for the lymphoma. Examples are the Onivax, which is being used for the adenocarcinoma, follicular adenocarcinoma, or small I11B10, which is being used for the lung cancer. So it is a antibody which produces a secondary antibody, and this secondary antibody can then act on the cancer cells and kill the cancer. So another types of vaccine is the cancer cell vaccines. Something like autologous or allogenic cancer cells were used to develop vaccine. These cancer vaccines are made from a person's own cancer cell or from the cell that are grown in the laboratory. These cancer cells are heat or radiation inactivated before use, and certain proteins of the cancer cells can be used for the cancer vaccine. Often cancer vaccines will also contain substance other than the cancer to boost our immune system, such as it can be combined with the adjuvant therapy like BCG. The main advantage of the tumor cell vaccine is that they have all the relevant tumor antigen needed for the immune system to mount the anti-tumor response. And a second advantage is that the tumor cell-based immunization allows the development of cancer vaccine even without knowing the antigen. So it's something like a pan vaccine, so that the tumor just take the tumor, either heat inactivate or radiation inactivate, and inject into the patient in combination with some adjuvant like BCG, and the patient will develop antibody against the tumor. And this is one of the most widely used vaccine that are in clinic, like like Oncovac, which is being used for the colon cancer, or Cancer Vac that has been used for the melanoma. Another vaccine is coming up recently, which is called the DNA vaccine. DNA from the patient tumor cells are injected into the patient, which instructs the other cells to continuously produce certain antigen, and these antigens are then they activate our immune system to produce the more T cell that ultimately help the development of antibody in our body against the tumor. So this is a type of another types of DNA vaccines. So if you know a specific antigen against a particular cancer, this antigen can be coupled, this antigen can be made a fusion peptide with a signal peptide, so that that can be released into the system, and this can be developed by transferring this gene into a viral plasmid and developing a virus that can be easily injected into the patient, and this patient will develop the antibody against the antigen, a chimeric antigen, and that can destroy the cancer. So this is a kind of DNA vaccine. So there are a number of vaccines which are in clinical trial, something like antigen vaccine, 5T4, CA, CMV vaccines, antigen-related vaccines, folate-related vaccines, epidermal growth factor-related vaccine, HER2-related vaccines that are being used for the different two different patients. There are also number of vaccines like human papilloma virus related antigen, mega antigen, mosetlin, MIP1, ESO, P53, PAP, or PSA. There are also and there are also the personalized neoantigen, something like if someone has a different types of cancer, they and this cancer cell develops a specific neoantigen that can be used for a DNA vaccine or a peptide vaccine, like mutated RAS vaccine, the RAS which are mutated at different positions that can be used as a antigen and develop vaccine that survive in telomerase, tumor-associated antigen, or WT1. So these types of vaccines are available, and there are other types of vaccines, that means the virus which develop cancer, retroviral which develop cancer, like HPV, or the HBV, hepatitis B virus, that were developed, that's Sir Jur Hussain, who developed the vaccine against the HPV, which causes the cancer into cervix, vaginal, or other anal cancer or genetic cancer. Nowadays, it is also known that the HPV can cause the oral cancer. Harold Jur developed the vaccine against this HPV, and for that he got the Nobel Prize in 2008. And this year, this hepatitis B virus, which causes the liver cancer, this was recognized this year by giving the Nobel Prize to Alter, Harton, and Rice, These three guys got Nobel Prize this year because of the discovery of this hepatitis virus. And this hepatitis virus anti-hepatitis vaccine was also developed, which cures number of which helps to prevent the development of the liver cancer, HPV-related liver cancer. So these are the latest development in the different types of cancer, and these vaccines, most of these vaccines can be given administered alone or in as with a adjuvant. There are a number of immune adjuvants available. So substances known as adjuvant are often added to the vaccine to boost their ability to protect anti-cancer immune response. Some microbes such as BCG can be used as a adjuvant for the cancer for the vaccine. BCG can act as a booster for the immune system so that the adjuvant, the cancer vaccine can work. If you have a boosted immune system, the body can develop better vaccine. The treatment of the bladder cancer with BCG is very well known. Treatment of the cancer can cause some symptoms like flu-like symptoms, with fever, but it can or it can cause fatigue. But sometimes it can cause a burning sensation to the patient. And there are a number of side effects for the cancer vaccines. Like the most commonly reported side effect of vaccine is the inflammation-related problem. Since the vaccines are injected in liquid form into the skin, they causes the redness, pain, swelling, and warming of the skin, itching, and occasionally sometimes they develop rash. People sometimes experience flu-like symptoms after receiving a cancer vaccines, including fever, chill, weakness, dizziness, nausea, vomiting, something like that. Side effect, which usually last for only for a short time, indicate that the body is responding to the vaccine and making the immune response as it does when the exposed to the virus. Other drugs can also boost our immune system so that the vaccine can be more effective. Something like thalidomide can be used to develop the for the treatment of multiple myeloma along with the cancer vaccine. Landilum also newer drug treatment for the multiple myeloma. BCG can be used for the uh, superficial form of bladder cancer, colorectal cancer, lung cancer, and melanoma. All these can act as a superantigen to activate our immune system, and this our immune system then can attack our cancer, which are in a mild form, and eradicate them. Recently, curcumin was also found to be a universal booster of the immune system and can act as a pan-cancer inhibitor. Medicinal mushroom and asparagus also used as a booster for the immune system so that they can act against the cancer. And the new paradigm came with the personalized cancer vaccine. So personalized cancer vaccine is something like you acquire the patient sample from sample from the patient, and this sample can go for the sequencing or analyzing or microarray to understand the identify proper neoantigen that can be targeted by individual cancer, and this neoantigen can be used to develop the vaccine, and then the vaccine can be administered. So depending on the nature of the cancer or individualized cancer, this personalized cancer vaccine can be developed, and these are now in clinical trial. Something like but, most widely evaluated is the advanced melanoma, and some also personalized cancer vaccines are in clinical trial, like brain cancer, breast cancer, colorectal cancer, liver cancer, pancreatic, and pediatric cancer. These personalized cancer vaccines are in clinical trial. So there are a number of challenges to developing in the field of cancer vaccines. One is the improving the how the vaccine can target or the identified of individuals, and optimizing the vaccine for formulation and the delivery method, and incorporating vaccine into the immunotherapy combination. So these are the challenge need to be developed for the development of the vaccine, proper vaccine. And recently, in 2018, the Nobel Prize came for the development of another types of vaccines, which is a part of immunotherapy, which is the the CTLA4 and anti-PD1 vaccines, which can inhibit the CTLA4 and PD1, which are the co-stimulatory molecule for T cells. And during cancer, these co-stimulatory molecules are overexpressed that can hamper our immune system. So by inhibiting this checkpoint inhibitor, you can boost your immune system for better immunotherapy or better vaccine therapy. For that, James Allison and Hozo got the Nobel Prize in 2018. So you can see a number of recent Nobel Prize came on the development of vaccine in cancer. So there is another, immunotherapy, which is a CAR T cell therapy. As you see, the T cells from the patients are collected, and that has been genetically modified to develop the CAR T cells. This is a chimeric antigen receptor. So the antigens that are present in the cancer can be identified and make a chimeric gene, and that can be inserted into the T cell, and these chimeric T cells then can be expanded into the laboratory to develop to get a high number of these chimeric T cells that can then be injected into the patient and cure the cancer. But the reality of this immunotherapy or vaccine therapy is that this is the real reality. So if you see, so these are the metagenomic data of all the cancers that were treated, all the recorded cancer over the world, all over the world, that has been analyzed, and this graph were developed. So if you look at, if someone has developed cancer and he doesn't go for the therapy, this person will die, definitely will die. 50% of the patients will die within 6 months to 1 year. So if you give them chemotherapy, you can extend their life from 1 year to 2 years. And sometimes, if the cancer is not that aggressive, some patients can even survive over the 5 years of over the. And if you give the genetically, if you go for the genomically targeted chemotherapy, you know, uh, the, the survival probability of this patient, they increase by several fold, because by the personalized cancer therapy, you can increase the survival probability of the patient. But if you go for the immunotherapy, the probability of the survival can increase up to 20%. 20% from the 5% to 20%. That is the advantage of the immunotherapy. So that after the immunotherapy, you can increase the survival probability over 5 years from the 5 to 7% to 20 to 25%. But the problem lies over here. The cost of the immunotherapy is huge. Not all the patients, they cannot go for the immunotherapy. So basically, nearly 70 to 80% patients, they don't respond to the immunotherapy. So we need to ask why these patients don't respond to the immunotherapy. Basically, before to understand why these patients they don't respond to the immunotherapy, you have to understand the tumor microenvironment. This is the picture of a tumor microenvironment. You can see there are different types of cells that do coexist in the tumor microenvironment. And interestingly, most of nearly 70% of these cells are immune cells. So there are a number of cells like macrophage, lymphoid cells, neutrophils, mast cells, fibroblasts, pericytes, endothelial cells. They do exist into the tumor mass. If you look at a tumor, nearly 70% of the tumor mass is not the tumor cells or cancer cells. They are basically the immune and other cells or stromal cells. So what are the role of these immune cells? That has been very nicely explained by Bob Schreiber in this pictorial graph. You can see this is a normal tissue or a normal individual when they are exposed to different types of carcinogens. All the times we are exposed to different types of carcinogens and that can transform some parts of our body cells into a transformed cells. These transformed cells can ultimately lead to a cancer. But as we have our very good immune system, our immune system can recognize these transformed cells and they immediately migrate towards the transform site and they produce lots of cytokines and other materials which can act on the tumor cells and kill the transformed cells. So we don't develop cancer. Sometimes this mutation is very smart and there is a sometimes more than one mutation and the cells which harbor these types of mutation they become very smart. What they do, they hide the tumor neoantigen inside their body, inside their cells, so that our immune system cannot see them. And so these tumor cells remain in our body in a dormancy state for longer time, sometimes more than 20 years, 30 years. And they always look for a opportunistic condition so that they can expand themselves. With the aging or by using of different types of antibiotics that can suppress our immune system, or during the organ transplantation, if you use the immunosuppressive drug, then these cells find a favorable condition. When they find that our immune system is very suppressed, they start proliferating very fast and they overpower our immune system so that our immune system cannot act against them. And that's the time when the tumor develops. So when we find a person is developing a cancer or a tumor, that means they have to go through this total pathway. And in that case, the patient becomes more, vulnerable to develop the cancer. So tumor, they have different way to develop the anti-tumor immunity, and our cancer, they also try to suppress our immune system to evade the immune system and for development. So by different way, they suppress our immune system and make the immune system, make the body system in favor of the tumor development. So the challenge for the chemotherapy or the immunotherapy is that, when there is a tumor cells, we use the chemotherapy, and during the chemotherapy, when there is a we apply when there is a development of tumor, we apply chemotherapy. We apply chemotherapy and we kill some of these tumor. And the very small number of tumors they survive from this chemotherapy. These are the drug surviving cells. these cells are the reason for the tumor relapse. Tumor relapse. Sometimes when we have the since we have a very good immunogenic cell, these immunogenic cells, they can target our this cancer if they can be protected by the immunotherapy or chemotherapy. These immune cells, they target our these drug surviving cells and stop the development of tumor relapse. Uh, but the problem arises over here during the tumor development. This tumor hijack our immune system and they make this immunogenic cell into a tolerogenic cell. That means these tolerogenic cells, they cannot target the drug surviving cells. So these tolerogenic cells, they cannot target our drug surviving cells. So these drug surviving cells, they are escaped from the immune regime and this the tumor relapse. This is the reason behind the development of drug resistance cancer or the development of the immunotherapy resistance cancer. So if we can use this technology, if you can understand who are making this immunogenic cell into a tolerogenic cell, we can stop them and we can use this as a adjuvant therapy to stop the development of this pro-tumor immune system. And if we can interfere by this system, we can again make our immune system into an immunogenic cell, so that our immune system now can target the drug surviving cells and we can stop the tumor relapse. So before that, we need to understand who are making the immunogenic cells. So lots of research needs to go to make this both the cancer vaccine or immunotherapy more successful. Thank you.
Yeah, thank you, Gauri,
Yeah.
for a
So,
interesting talk. And there are questions,
thank you.
and I'll start with the first one. I think that was on your dinosaur with the Lancet Oncology paper, which came in August. They want to know how was it done? Was it carbon dating? So this question is from Michael Durairaj. The 72 million years ago calculation of first known cancer, it's based on which method? Radiocarbon 14 dating?
Yeah, I think they did, no, actually they did the skeletal scanning and found that there is a tumor, skeletal tumor in the brain. That was the evidence. That was the evidence.
Right, but they
They
used...
didn't. Yes.
Right. Then there is a question from... Yes?
Yes.
There is a question from, another question from Ms. Arya. How can we be sure that anti-cancer vaccine will not raise autoimmunity, or how this problem can be addressed?
Yes, that's a very good question. Sometimes the anti-cancer vaccine, they stimulate our immune system, but the problem is that during the if it is a prophylaxis, that can cause the sometimes the activation of our immune system. But if it is a curative or therapeutic, it cannot, because by that time when we administer the anti-cancer vaccine, our immune system are really automatically suppressed, because the tumor, they mask our immune system and they suppress our immune system by developing number of differentiated cells like Treg cells, gamma delta T cells, tumor type 2 macrophage, myeloid derived suppressor cells. They always suppress our immune system. So there is no questions of developing autoimmunity in therapeutic. But definitely in prophylaxis, there is a possibility of developing the autoimmunity during. So before giving the therapy, prophylactic vaccine, you have to be very careful so that you don't develop the you know autoimmunity.
Yeah. There's another question, and you're going to like this. Isn't curcumin also used for breast cancer? If it is
Yes.
used as a natural prevention for breast cancer, then how is it used for pan cancer?
Yes, the curcumin can be used for colorectal cancer. First, it was used in clinical trial for the colorectal cancer, but it can be used also for brain cancer and breast cancer. Curcumin acts in two different ways. It can act as a double-edged sword. In one hand, it interferes this is a multi-kinase inhibitor. It can inhibit number of kinases that can stop the proliferation of cancer cell. It is a p53 activator, so that it can induce apoptosis of cancer cells. So it can reduce the tumor mass. And other way, it can activate, it can rejuvenate our immune system from the tumor mediated immune suppression. So this rejuvenated immune system can can boost the vaccine development. So this pan-caspase curcumin, a pan-cancer inhibitor curcumin can act as a dual system, dual activator for the cancer.
Then there's a question from Rajeshwari Parida. Does these cancer vaccine provide prolonged immunity, thereby generating memory cells? What's the success rate of these vaccines so that the patient doesn't get to grow the same cancer cell again throughout the lifetime?
Yes, most of these cancer cells, most of the vaccines, except some of the viral vaccines, they are incorporated, they have a long lasting memory, because these antibodies are developed in the germinal center. So where there are number of memory T cells or memory B cells that can remember this antigen, and whenever you are exposed to in case of virus or bacteria, even the transform cells, same types of transform cells which develop different similar types of neoantigen, this vaccine can be they can develop again antibody.
Yeah, but Gauri, I think I would contradict
this is a long-lasting vaccine.
but we still do not have enough data from cancer vaccines to talk of long-lived memory.
Yeah, but the first cancer vaccine, which was developed in 1970, they found that with the repeat exposure for this cancer.
No, in the animal studies, but in the human clinical trials, we don't have. We just need more data.
Actually, only 5 or 7 years back, only 2000 years, these cancer vaccines are in clinical trials. So it's not yet the time, but to see whether it has a longer memory. But in mouse model, it was found that it has a long memory.
Right.
In clinical, in preclinical trial, it was found.
Then there is the question of peptide vaccine. Mode of action of peptide vaccine, how is it different from heat inactivated or attenuated vaccines, or a bigger vaccine molecule? And this question is again from the same person, Michael Dolaraj.
Generally, heat inactivated vaccines are developed with the whole tumor mass, or the UV radiated vaccines were developed from the whole tumor mass. So it has number of proteins that can also cause the autoimmunity in our body, because body's other proteins, because the tumor, they also produce the similar types of protein which are produced by our normal body cells. So they can have a non-specific antibody that can cause problem or autoimmunity in our body. But if you are going to target a peptide vaccines, depending, designed upon the new antigen that are developed by the cancer cells only. Then if you develop any antibody against that, that will not harm our body's other cells or somatic cells. So this is more specific for cancer than the heat irradiated or attenuated vaccine.
Yes.
So peptide vaccines are coming very fast, and you see the mode of the use of these personalized cancer vaccines is depending on the specific new antigen that are derived from a person's person, because most of all the cancers from person to person are different. So this from the peptide vaccines are now modified to personalized vaccine,
Yes.
which is coming in the next generation.
Yeah. Then there's a question from Ankita Ghosh. Has the vaccine been developed that can target cancer stem cell?
Yeah, like survivin vaccine, the vaccine against a protein called survivin, which is really required for the for the transformation from a cancer stem cell to cancer cell, because cancer stem cells itself is not dangerous, but they can produce cancer cell. They can differentiate, multiply and produce cancer cell that causes the problem, because the cancer stem cells are very small number. They don't expand into high number unless you artificially change their function. So cancer stem cells, when they proliferate, immediately they differentiate into cancer cell, and this cancer cell proliferate highly to develop the tumor mass. So if you can target the cancer stem cell by using the anti-surviving peptide antibody, then you can stop the cancer stem cell development, so that these cancer stem cells, they don't survive, and these cancer stem cells are really the drug surviving or drug resistant cells. So once if you can target these cancer stem cells, then the chemotherapy can tackle the rest of the cancer cells. So if you can combine the cancer stem cell therapy along with the chemotherapy, you will be able to eradicate cancer better than the chemotherapy or the immunotherapy alone.
Then there is again question on whether MUC1 peptide vaccine can become an effective vaccine and how. That's again from Ms. Hina.
Can you repeat that?
Can MUC1 peptide vaccine become an effective vaccine? And if yes, how? MUC1 in breast cancer.
Actually,
it's a tandem repeat of
yeah. I know that. I don't know really the science behind that. I'm sorry. Yeah.
I think it means we still do not, as I said, this field is still new, and animal experiments have shown that MUC1 peptides generate T cell responses, but in clinical trials, I don't know how this has not been.
No, I don't have any clue about this. Yeah. I don't have clue of some of these because this is a review. I presented about the vaccine to give a general idea about the vaccine. Some of the details of some of the vaccines I don't know really. Just to give you an overview, I mentioned about this vaccine.
Yeah. Then is Gauri Shankar there? Not Gauri, I mean Girdharira. There is a question for him also.
Yes.
Yes. Then the questions for you from Uma Kanga. Classical HLAs are downregulated tumors. Is there any role of non-classical HLA antigens in tumor evasion?
Right. So that's very interesting question. I can tell you the way tolerance is generated in the body. The best example, if you look into nature, is the pregnancy. In pregnancy, you have at placenta a lot of non-classical MSC molecules, and when these cells interact, the immune cells interact with them, they do regulatory function. So now, if you take the example of tumor, where you again develop tolerance to the tumor anti-tumor cells, so they do actually contribute in regulatory phenotype non-classical MSC molecules. So this is the one way actually. It's known that even in gamma delta T cells, they do interact with these classical, non-classical MSC molecules, and they contribute in regulatory phenotype. Yes.
Or I can just add to that your NKT and CD1D.
That's correct. Yes.
That's a classical example.
Example of yes.
Then there is something again on carbon dating. Somebody had asked something, but there's something on. Put some light be shared on phototherapy, including photothermal therapy and photodynamic therapy.
So very good question. What I showed you in the circle of immunity, that tumor immune cycle, in any way, either by chemical or by physical method. And the phototherapy is one way physically you damage the cells and you expose your tumor antigen in the surrounding. And whenever tumor antigen come out of their territory, immune cell sees it as positively as and mount the immune response. So the abscopal effect, one of the known phenomena in tumor and tumor metastasis, that yes, if you do this localized phototherapy on a tumor mass, you do develop anti-tumor immunity, and that's called abscopal effect, and that has a very important role in mounting the anti-tumor immunity. Yes.
Then either of you can answer from Dr. Khan that can we make ACE2 homologs molecules to be used in the therapy for COVID-19?
Okay.
There are a number of attempts are going on to there are AC homolog structures are being used to stop the binding of the ACE with the RDB, receptor binding domain of COVID-19. Yes, there is one paper published in Biotics recently. They tried to find out a number of natural compounds that can stop the binding of the RDB with the ACE2 because they have a similar structure homology.
Now I'll take the last question, which I'm intrigued by the way it is asked. Some say this organ or system-based approach is wrong. Like tap is leaking, bathroom leak, kitchen leak, sink leak. It's basically leak. It needs to be addressed. Would you like to comment?
What?
Is it directed to me or Dr.
No,
Gauri?
it's to both of you. Anyone can
Perfect.
ask.
So when there is a leak and there is a damage, and damage is not good for anyway. You require a perfect shape and then say that, your bathroom is okay, can I use it? And use it in correct way. So when your system is leak and damaged, you don't expect that your immune system will be working fine. Only the correct system will give you the correct output. And when we talk about leaky, referring to the leakiness of what? Leakiness of antigen coming out of this, leakiness of specific immune cells coming out or going in. If you're talking about that, so if molecular leakiness is you're talking about, yes, it do contribute to it. And if you say that it's a physical leakiness, then I think then the whole thing is not going to work. You require correct shape in order to have the correct response.
Yeah, this was from Michael Durairaj. Okay.
Yes.
So now I'm going to take the last one. Does use of excess of immune boosters lead to cancer?
Yes, definitely it does. Because one of the example is inflammatory bowel cancer. That's because of the overactivation of our immune system into the bowel system may lead to the colon cancer.
Yeah. So I guess we've addressed most of the questions, and it has been a very interesting session. I thank both of you, Dr. Girdharilal and Dr. Saab, for making this an interesting session. The topic is really very vast to be covered in 2 lectures, but you all did a wonderful job. And I thank you, and I thank the entire audience who are on the web today listening to these talks. And of course, the organizers for organizing this program. Thank you very much.
Thank you.
Thank you.
Thank you, madam,
Thank you,
for
Subha.
this very
Thank you.
Subhadhi. Thank you,
Subhadhi.
Subhadhi.
Yeah. Thank you,
Okay.
Amit, and thank you,
Thank you, Sunil.
Sunil.
Thank you,
Yeah.
Girish.
Thank
Yeah,
you.
bye.
Hey,
Bye.
Amit, how are you?
Finally, we
Alright.
can see him.
Mike,
Yeah.
Mike, Amit, your mic is muted.
All
Hi,
right.
hi, ma'am. How are you? Hi, Girdhari. Hi,
Hi.
hi, my superstar, Gaurida.
Yeah. So-
Girdhari, your background, you're sitting outside of the institute, or you? What is the thing?
No, I think this is what the digital things you can do whatever you feel like.
Okay.
So
Wow.
I'm actually in the lab.
Right.
Sunil, you must
So-
teach us how to do that.
Hmm.
So that
Yes,
Shiva
now
can
you
make
can see I'm outside the lab.
background.
The magic is
Shiva
here.
can make his her background
This
like
is
the
more
Yes.
I'm
Indian Ocean.
I'm at my home background.
Great
So
talk.
when are we
So
meeting
we
again?
make
You see, Girdhari's original background.
Yeah.
That looks more usual.
Yeah.
Right. Yes.
Okay, Girdhari, thank you very much. Gaurida, thank you.
Thank you.
And
Thank you, Sunil.
very
Thank
nice.
you.
Only you made it very nice.
Yes,
So,
yes.
Yeah.
That was good.
After
That was
lots
good.
of
That we were
problems. After lots of problems.
Yeah, we were taking as
Actually,
many questions for Girdhari to
the
answer.
actual the
Oh,
problem
that's
is that
fantastic. I think
Mac
they all are nice questions.
Yeah.
Mac
So
doesn't
Girdhari carried on and on like a batsman. He was not going till you came. So it
Yeah.
was very nice. He answered so many questions. Thank you very
the
much.
problem with the Mac, it doesn't allow lots of features of Zoom.
Huh.
Google
Mac
Meet
No,
is
actually,
fine.
yeah.
Google Meet is fine for the Mac, but the Zoom doesn't fit good
No,
in
Zoom,
Mac.
you have to give the audio as video access in the security
Yeah.
privacy.
No, I
setting.
did, but still, I can hear my voice, but it is not being transmitted to the Zoom. So that's why I prefer Google Meet better than the Zoom, because this is a. I
So-
don't want to discuss.
Sunil, what time are we again meeting?
5:45. So
5:45.
session starts at 6. So 5:45. We all break now. Have some tea and all, and get ready.
Yeah. Yes.
Okay.
Sure.
Okay.
Bye.
Thank you. Bye.
All right. Thank you.
So Sunil, you are sending the tea to us.
Virtual.
Yes.
Yes, really hot tea.
Yeah.
Yeah. Sure. Okay.
Okay.
Thank you.
Okay.
All right. See
Okay.
you.
See you.